So welcome again. It's very nice to see a full room. We have 33 people here with us watching. So I'm Ali, and you can see Meryem Beklioğlu as well. We're the co-organizers of this. So we're gonna, for those of you that just arrived, I'm gonna give a brief overview of each of the presentations by sharing my screen. And then, so during that time, I'll ask you to turn your cameras and microphones off. But then after that, we'll open it up for the question and answer session. And then everybody can feel free, please open your cameras and mics, and we'll just see where it goes from then. And hopefully we'll have a nice exchange. So I'm gonna share my screen now. Can everybody see?
Yes.
Yes.
Yes, Ali.
Okay. My internet is kind of freezing. Okay. Well, I'm going to assume that you're seeing. So our session was
Ali, you can turn off your camera as well. That will make it easier because we are looking at your screen. Yeah.
That's right. How do I do that?
Just turn off your camera.
I have to find it first. Okay. All right. Can everybody hear and see?
Yes, it's fine.
Okay. So the title again of our session was Linking Functional Diversity with Ecosystem Function Across Landscape Scales. And we had some talks that were from landscape scale, some talks that were not. We had some talks that linked functional diversity or functional traits. This is actually the unupdated title. It should be Linking Functional Traits with Ecosystem Function. Anyway, some talks fell more or less, but we joined them anyways. So hopefully in the discussion, we can tease out how to link these two concepts. So the main objective that we want to stimulate, the discussion that we want to facilitate, is how can we link these traits? Everybody's talking about functional traits and their diversity. It's increasingly popular. But how do we link these with ecosystem functions like energy flow or nutrient cycling, for example? So there's a lot of different kinds of traits, but usually we focus on very few of them, like size, for example. So I see that here we have some different kinds of traits, which is really exciting in some of the talks. And we have four main questions that I just want to remind you when you're thinking about your questions and also when we're thinking about the discussion. So the first one is, can functional traits be used to predict ecosystem function? We don't even know if that's possible. The second question is, what's the role of traits on different functions like stoichiometry, respiration, or trophic transfer efficiency? Do specific traits correspond to specific trophic interactions? Like, can we base on, if there's a larger zooplankton size diversity, does that mean anything about some kind of trophic interaction, for example? Hello?
I will hear.
It's okay, Ali.
Yeah, continue, Ali.
And the last question is how to link pattern with process using a trait-based approach, right? Because most of the time we either know the pattern, like we do a monitoring study, we know the community composition, for example, or abundance and biomass, but we don't know the process. Or we measure the process, like respiration or grazing, but then we don't know how that applies in nature. So can we use a trait-based approach to link these two? So I'm just going to follow the same order that the talks were organized on the website. And we'll start with Miloš from Serbia. And his title is Application of TBI Index in the Ecological Status Assessment of the Shallow Aeolian Lake Ludaš. And the main idea here was to test the applicability of a recently developed diatom index in ecological status assessment for degraded saline wetlands. And they focused on a single saline lake, if I remember correctly. And their hypothesis was that the epiphytic diatom assemblage sampled from macrophytes in saline habitats will contain diatoms with different ecological preferences and functional traits compared to soda wetlands in natural status. And their main results I tried to summarize here. So here we can see the index on the y-axis and then the lake that was studied. And they compared this index, which had a lower value in this Lake Ludash, compared to what they considered healthy lakes. And that one of the main results that I noticed, which related to traits, was that the non-motile, more slender diatoms were indicators of healthy systems. Then we had Rosenberg's talk from Brazil with the title "Changes in Plankton Size Diversity are Driven by Both Bottom-Up and Top-Down Control in Semi-Arid Reservoirs." And they hypothesized that nutrient enrichment reduced the size diversity of phytoplankton and zooplankton. And they also hypothesized that omnivorous fish size diversity affects zooplankton size diversity more than phytoplankton size diversity. And they compared among mesotrophic and eutrophic systems. So they answer their questions here. For the mesotrophic systems, nutrients did reduce plankton size diversity, but in eutrophic systems, it did not. And then the second hypothesis about omnivorous fish size diversity, whether it reduced zooplankton size diversity, it depended on the zooplankton size range for both eutrophic and mesotrophic system. So they conclude that plankton size diversity affected by bottom up and top down, depending on the trophic state. The next talk was from Melissa from Turkey, and her title was "Combined Effects of DOC, Dissolved Organic Carbon, and Zooplankton with Contrasting Traits on Phytoplankton." Her hypothesis was shown here graphically as well, that the DOC would reduce the phytoplankton biomass. It would increase the proportion of mixotrophy. There were two kinds of DOC in this experiment. One was more recalcitrant, one was more labile. So they would have different effects on the phytoplankton, the labile having a less negative effect. And that grazers, depending on their traits, would reduce phytoplankton, some grazers more than others. And they found that DOC effects varied on the DOC source and also according to the phytoplankton species. So in the lab experiment, they showed that mixed DOC decreased all species, but there were some species-specific differences. And in a mesocosm experiment, they found that the more labile DOC increased, but the recalcitrant DOC decreased phytoplankton. And for the grazers, they had negative effects on the phytoplankton, but it depended on the traits of the grazer and the phytoplankton species. The next talk is by Sinem, also from Turkey, and the title is "Effects of Dissolved Organic Carbon and Zooplankton Grazing on Bacteria and Ciliates." And her hypotheses were graphically illustrated. So in this case, she thought that the DOC would positively increase the bacteria and ciliate biomass, and the grazers would decrease bacteria and ciliate biomass. And again, there would be differences according to the traits of the grazer in this effect. And in the mesocosm assay, grazing assay, the hypothesis was that the labile DOC source would have a positive effect, but the recalcitrant would not have any effect compared to the control, and that a mixed grazer community would still reduce the bacteria and ciliate biomass. And the results for the laboratory study was that there was little effect of DOC of grazers on bacteria, but DOC did increase the ciliates, and grazers reduced the ciliates. So grazer traits regulated the ciliate biomass more than DOC. And in the mesocosm assay, there was little effect of DOC on or grazers on bacteria. And the leaf leachate, the more labile DOC, increased the ciliates. The next talk is by Meryem from Brazil. and the title was Flower and Leaf Litter Interaction Promotes Non-Additive Effects in the Aquatic Environment. And they evaluated if and how the interaction of labile and refractory litter with different proportions affect decomposition rate. So they had flowers and leaf. So the flowers were more labile and the leaves were more recalcitrant. This is interesting because in the previous talks, the leaves were the more labile one. So I thought that was interesting. Anyways, they hypothesized that the effect on decomposition rate would be dependent on the proportion and abundance of the flower litter, the more labile litter. And they found that the results supported this. So there was higher decomposition for the flowers, and they suggest that flowers are a biochemical hotspot for decomposition in shallow lakes where there's a lot of flower litter. The next talk was from Wojciech. I'm pretty sure I'm destroying your name. I'm sorry about that. The title was Nutrition Facts, Impact of Cyanobacterial Blooms on the Carbon to Nitrogen Ratio of Freshwater Crustacean Zooplankton. I was able to find a picture for everybody except for you, so you're the mystery person here. And the aim here was to examine the effect of cyanobacterial blooms on the nutritional value of planktonic crustaceans in shallow water bodies across a latitudinal gradient in Europe. And the hypothesis was that group-specific nutritional value of planktonic crustaceans is modified by the latitude effect in shallow water bodies with blooms occurring. And they found that for cyclopoid copepods that there was no difference in the C to N ratio in any of the latitudes. And the calanoid copepods had some difference. They had a lower C to N ratio in northern latitudes, and Daphnia had a higher C to N ratio in the northern lakes, and Diaphanosoma species had a higher C to N ratio in southern lakes. So 3 of the 4 zooplankton groups studied had a latitudinal response. Our next presenter or would-be presenter was Nathan from Brazil as well, and the title was Phosphorus Total phosphorus and pH are key variables for functional structure of zooplanktonic communities in an agropastoral landscape. And the objective here was to understand the influence of agropastoral landscapes on ecosystem functioning, especially through a study of abiotic drivers on zooplankton diversity. And they concluded that total phosphorus and pH were key variables for the functional structure of zooplankton, and that especially alkaline waters and preserved environments provided a more complex functional structure in zooplankton. Our next speaker is Evaldo, and he was in Brazil, now he's in the United States, and the title is The Importance of Copepod Grazing and Cyanobacteria Defense Traits on the Top-Down Control of Cyanobacteria. So the objective was to evaluate the effects of copepod grazing on the facilitation of cyanobacterial dominance in 2 experiments, one with a single copepod over microcystis, and another with 2 different copepods, a calanoid and a cyclopoid, over the filamentous Cyanobacterium haphidiopsis, which used to be Cylindrospermopsis. And they found that facilitation happened with microcystis, but not with haphidiopsis. And that the active feeder, so here's the trait of the calanoid, had a stronger grazing impact than the cyclopoid, the raptorial cyclopoid. And then we continue with Lorena, also from Brazil. Her title is Trait-Based Zooplankton Community Assembly and the Functioning of Aquatic Ecosystems Along a Productivity Gradient. And she asked, how do zooplankton body size, diversity, and composition respond to a gradient of nutrient addition? If this response is modified by the presence of artificial plants, and what is the relative importance of community average size and size diversity in determining top-down control of algae? and what's the relative importance of trait changes in response to nutrient addition versus the direct effect of nutrient addition in determining top-down control. So they found 3 key results. The first was that increasing nutrients had a strong and positive effect on the size diversity and community mean size of zooplankton. They also found that changes in zooplankton size structure beyond just richness mediate the long-term effects of nutrient enrichment and top-down control. And they suggest to maintain, in order to maintain high levels of ecosystem functioning, such as strong herbivory, it's important to maintain high body size diversity, as well as a average mean size. Then we continue with Vildan. She's from Turkey, and her title was Phytoplankton Community Assemblage Processes in Turkish Shallow Lakes Using Different Functional Diversity Components. And Vildan's only picture that I could find was with her back faced, but at least I found a picture of her. And it seemed it was more like a work in progress. So they had some expectations, the classical expectations of meta-community theory, right? Dispersal versus connectivity. So if environmental filtering versus dispersal is controlling the community assembly. And they found that environmental filtering was more important than dispersal, and especially the relationship of phosphorus and nitrogen with functional diversity, though they still have some work ahead. So maybe it would be nice to receive comments and suggestions for her. And then finally, it's yours truly for Brazil. And the title of my talk was Zooplankton Grazing Mode and Body Size, Predict Ecosystem Function Across 16 Tropical Lakes. So we wanted to compare this relationship between the trait diversity of zooplankton, the identity of traits, and the resource use efficiency, which is a proxy for top-down control. So we hypothesized that there would be some correlation between trait diversity and resource use efficiency. Then we also hypothesized that the trait identity would be a stronger predictor than trait diversity. And among the different traits, we thought that grazing traits would explain more of the resource use efficiency compared to mean community size. And we found that the first hypothesis was did not occur. So trait diversity was not that important for resource use efficiency of zooplankton. We found that trait identity was more important. And among those traits, we found that the grazing mode or behavior was more important than mean body size. So I'm sorry that I did this to your presentations, and I want to congratulate all of you for putting it together and sending it to our session. So now I think we can open it up to questions. I would just like to see here, so I'm on the website and I can see that there are 4 questions for this talk of Wojciech. So let's see what these questions are. Okay, is everybody hearing me still?
Yes.
Yes.
Okay.
Yes.
Okay. So the first question is by Maria Caldero Pascual, and she's asking for this talk, Nutrition Facts, Impact of Cyanobacteria Blooms on C to N ratio of zooplankton. So she says, hi, congratulations on your interesting presentation. And I have a technical question. I'm trying to carry out grazing experiments with Diaphanosoma and would be interested to know how to manipulate them. I have seen that it is one of your studied taxa. I have also sorted them for, I don't know what these things are, fatty acid and CNP content, but when I try to keep them alive, I am finding it very tricky. They are soft or seem to be trapped in air bubbles. Due to pandemic restrictions, I haven't been able to try that much and only appearance during summer. So just trying to be ready for summer. So she's asking if there's any trick that you know that you might be willing to share to keep Diaphanosoma alive.
Hello, Ali. Thank you for presenting my presentation once again and answering the question of Maria. Just like I replied in the comment in the platform of the conference, I have a limited experience with the culturing of Diaphanosoma and other plankton crustaceans. My tip for her is to use some pipette and isolate the material from the dense samples collected in the field and keep transferring the material to the next drop of the water and try to select another one time just to proceed to the situation when there's a drop of water with the single specimen of the species of the interest.
So you don't do anything special for Diaphanosoma that you do to others?
Actually, I think that the Diaphanosoma is maybe similar to culture like Daphnia. So they just need some food, for example, Cyanobacteria, maybe some yeast, and they maybe process just like I said. I think that using the paintbrush, like Maria stated, or the needle may actually hurt the Diaphanosoma, and they may not perform as well as before the procedure.
Okay, and I see now that you already answered. I wasn't aware of that, but I think it's good for everybody to see here. So the next question is from Carolina, and she says that she did not understand why a higher carbon to nitrogen ratio would indicate a better food or nutritional quality, because usually when the C to P ratio is lower, the quality is considered better.
The assumption of our study was that the carbon to nitrogen ratio is correlated with the lipid content in the organism. And we tried to track the changes of the carbon to nitrogen ratio in the studied organisms. And also the lipid content is a proxy for the fitness and the stored resources inside the organism, and our hypothesis assumed that the latitudinal gradient may connected with the cyanobacteria blooms may be a factor driving the loss of the carbon to nitrogen ratio, and as an effect the worse condition of some of the studied animals.
I see. So it depends on what kind of quality metric is being used, maybe the results would change.
Yes, definitely. And also the driver of some changes, including the carbon to nitrogen ratio, might be some functional traits, including the selectivity of the food and some other traits, just like size of the body of the organism, which might be also affect the trophic interactions, the freshwater food webs between the plankton crustaceans and the cyanobacteria.
Okay, thanks. So we might come back to your talk, but I just wanted to go through these questions on the website so that I can close this, stop sharing my screen, and then we can have a more normal question and answer.
Okay, thank you very much.
Thank you. So the next question is for Miloš's study, the application of TBI index in the ecological status assessment. And the question comes from Sinem. So she thanks you for your presentation, and she has a question about the ecological group slide. So you explained that blue bars represent good conditions and yellow bars as planktonic. I'm confused about how can we compare these groups. Does planktonic mean that there are poor conditions? Thank you.
Okay. Thank you for the question again. And as you can see, maybe on the model, I have already answered the question, so I will just make some kind of a summary. I'm aware that this research is not connected so much to ecosystem functions, but it's connected to conservation of these fragile ecosystems. So for us is very important that we can use this functional approach to protect this ecosystem efficiently and to offer a tool. So this threat-based index is hard to say formulated or constructed by using two groups of ecological groups of diatoms. So the first group is how to say positively correlated with conductivity, because saline wetlands, or those soda pans as we call them, are endangered because fresh water usually degrade them and used by human beings or us, because they are in the vicinity of agriculture area. So those diatoms that are positively correlated with conductivity or salinity, they are indicators of a good or healthy ecosystems. While on the other hand, we have diatoms that are related to fresh water, or they are negatively, statistically negatively correlated with conductivity. And among them are planktonic diatoms, because usually in fresh water that comes from the rivers and enter into the soda pans, we have plankton diatoms. And this index is very good in that sense that it is sensitive to the presence of those groups. So it's basically the answer to this question. And I have just one remark. You mentioned in a short introduction of our research that non-motile slender diatoms, that it's a mistake, not non-motile.
Oh, sorry.
Motile.
Sorry.
Yeah.
Yes. Sorry.
No problem. Yeah.
I apologize about that, which is a shame because I actually know that the more slender ones have the necessary morphology to become motile. But anyways, so I actually had a similar question, but I think the confusion here about Sinam's question might be because what you consider a degraded system is simply a more freshwater system. Is that correct?
Yeah, it's correct. And and there was one more thing about that. Soda pans are usually hyper-eutrophic waters. It's normal to have a very, very high concentration of phosphorus in them. So everything is opposite to freshwater ecosystems. So we cannot apply those measures or some kind of a biological quality elements from the Water Framework Directive for fresh waters, because everything is upside down. So we need a new tool to assess the degree of their degradation of those ecosystems.
Okay, thank you. So now we can just open the floor to questions in general. Maybe just to keep some order, we can go from the same, we can just continue the order that was on the website. So if anybody has more questions for Milos, we can take them. Or I don't know, Meryem, do you think there's a better way of doing it?
This is good. I think we can, since we gone through the questions already asked, now we can open the discussion to all the participants to this session now. Yeah.
Okay. So I'm just checking the chat now to see if there's any questions. Nestor has some.
For you.
But let's try to stick. Okay, Lorena has one for Milos. Great. Let's start with Milos, and then we can go from the same order of the talks. Go ahead, Lorena.
Hello to all. Thanks for presenting my main results, Ali, and thanks for your presentation, Milos. I just have one question, and it's about the range of the index that you used, TBI index. What's the range of this index?
Thank you, Lorena. I think I mentioned in my presentation that the range of index is from 4 to 9. And I just want to say that index is made by a Hungarian group, Professor Cila of Stenger Kovacs, and they published a paper. I think I put that paper in the reference cited in my answer. So if soda pen has 9, it means that it is in natural or a reference status. And if it's approaching the lower limit, it's degraded. So we are now testing this index in 7 soda pens. Actually, our publication is accepted the last week. We compared 2 degraded and 5 soda pens in natural status. And we see that this index is promising tool.
Thank you.
You're welcome.
You're muted.
Any more for Milos? Okay, so let's move to the next one, which is what? I have to go back to the website here. But if anybody knows, they can say already. So it's changes. It's your talk, Rosenberg.
Yes.
Did anybody have questions for Rosenberg?
Hi, Rosenberg. It's Evaldo here.
Hey, Evaldo.
I have a quick question. So I was wondering if, I know this might be a tricky one because it's like we're all talking about latitude, same latitude. So I was wondering if temperature might deviate between your two regions. When it comes to like, I was wondering if the area of the lake might play a role in that, because if you have smaller lakes, then you have like temperature changing more abruptly during like daily cycles. And if that may play a role in the traits that you're observing, and if there is like any small difference, even if you've observed something between the two regions that you studied. Because we just don't talk about temperature because we're only talking about like, you know, like your latitude is very small, but I was wondering if because they're in different like areas, it might play a role in the biome that they're inserted in.
Well, thank you very much for your question. It's really nice to be here, even though we are a little bit far from each other. But I'm happy actually to be taking place of the Shallow Lakes Conference, even though it's we are, you know, in a remote mode, let's say. Well, Evaldo, thank you very much for your question. It's good to see you as well. And well, when we decided to perform this work, it was very clear for us that temperature will not be an important variable to explain differences, for example, in the traits of zooplankton, phytoplankton, fish. Because all the two basins, if you remember, they are inserted in the same area in Brazil. The main differences between these two regions are precipitation. And because of the precipitation, there were some differences in the trophic status of the reservoirs as well. So we don't think, well, of course, we know that temperature is a very important environmental variable to change traits in the zooplankton, phytoplankton, and fish in food webs. We know that. It's very clear when you check latitudinal gradients that you have some effects on the size of the organisms. But of course, the size is also influenced by predation, for example, and other environmental, biological effects that may somehow have some influence on the structure and the size structure of phytoplankton, zooplankton, and fish in these reservoirs. But we do not see this. I think I hear something.
Yeah, if we can turn off our microphones who are not talking.
Okay, sorry for that. So I don't think temperature is a very important trait to change size structure of phytoplankton and zooplankton, at least in the kind of systems that we are working, Evaldo. I don't know if I answer your question, but definitely. I don't think temperature is important for us.
I just had to ask because, you know, because I was curious.
No, it's okay. Thank you.
Thank you.
I have a question if there are none others. So if I understood correctly, we can expect the top-down effects of fish on plankton size diversity to depend on trophic state. Is that correct? So
yes,
why do you think that is?
Well, these lakes, they are slightly different when we talk about trophic status. You may have noted that I named the lakes as less eutrophic and more eutrophic. I didn't want to use mesotrophic, Ali, like you added in your presentation, because you are not mesotrophic. Both groups of lakes are eutrophic. But when you check the total phosphorus rate, when you compare the rate of total phosphorus in the lakes that are inserted in the region that rains less, they are completely different. The concentration of nutrients are different. Let's say that we had a group of lakes that were hypereutrophic and a group of lakes that were eutrophic. But we cannot say that the lakes that were inserted in the region that rains less was mesotrophic. But well, I will answer your question about the top-down effect. Even in tropical regions, when you have lakes that have more, that have less nutrients and they have higher water transparency, you find a fish community structure that is different for those lakes that are more eutrophic because of the water transparency. So the lakes that are less eutrophic, the water transparency was high. So we had a higher proportion of piscivorous or omnivorous fish compared to the lakes that were more eutrophic. So when you have this kind of structure in the food chain at the top of the food web, you'd have some effects on the size structure of the zooplankton because you decrease the abundances of omnivorous plantivorous fish. So this somehow relaxes the effect over the zooplankton size structure. So you somehow would have a more diverse group of zooplankton, for example. I mean, the effect is not different for what, I mean, not the effect, but the pattern that you see in temperate regions, for example. It's not different for what we see in tropical regions. So that's why we found some differences in the effect from the top to the bottom of the food chain in our work, at least that what we think that is taking place in the size structure of the plankton communities in these reservoirs.
I see. Okay, thanks.
I have one question.
You're welcome.
To Rosenberg. I don't know if I understood well. You found an addition of large zooplankton, right? large zooplankton species with nutrient increasing. Is it right?
Yeah, actually, we divided the zooplankton in micro-zooplankton and meso-zooplankton. They are not large like those ones that we find in temperate regions. If you check the size range of the zooplankton, the maximum size is going to be 1,200 micrometers, for example. It's not a large zooplankton. No, but yeah. I decided to divide the zooplankton range sizes because I wanted to know if, for example, the variance of size diversity of omnivorous fish would have different effects on the size range of micro-zooplankton and meso-zooplankton. And that's what we found. Even though the total phosphorus had a positive effect on the size structure of the zooplankton, meso-zooplankton, like you said, the relative importance of size diversity of fish was higher than the nutrients.
Yeah, so, yeah, no, this is interesting because I and my co-authors demonstrated the same result using experimental design, but without the fish pressure. So what we found it was that in fact phosphorus increased the size diversity and also the community average size of zooplankton. And when I see your results, I think, oh, okay, this is it. This match with what we found, because you include the fish pressure, fish predation, and we did not include this factor in our experimental design. So it was fantastic. And just one question, what are your thoughts about the mechanism that drive this observed pattern that you found in your study?
But can you be more specific?
Like the mechanism that allow for small and large species be in these systems, in this eutrophic system, like just the fish pressure. Do you think that maybe the diversity in resource may be a factor that allowed this?
Yeah, definitely. Yeah, the diversity of resources would definitely have some impact on the size structure of zooplankton communities. Actually, if you see, I did not draw attention on the cyanobacteria, but if you take a look at the cyanobacteria results, you see that actually there is a very strong effect of cyanobacteria on the size diversity of zooplankton as well. The mesozooplankton is actually benefited by cyanobacteria in eutrophic lakes. Nice. So even though you have an impact from the fish, you also have an impact from the bottom as well. So yeah, it's very interesting, but we still need some more analysis, let's say, to try to disentangle how
yeah, exactly,
how important it is, for example, the diversity of resources to explain the variation, the size diversity of mesozooplankton and microzooplankton as well in these reservoirs.
Yeah, and it was a positive impact of cyanobacteria?
Yeah. Yes, oh, yes. When you increase the size diversity of cyanobacteria, you actually have an increase in the size diversity of mesozooplankton that is comprised mostly by copepods, for example. But yeah, Ali is going to talk a little bit
about I know. Okay, perfect. Thank you.
You're welcome.
I think Ali has frozen. I'll take over. Any other question? It's good to have spare co-hosts. Any other question to Rosenbergs? If not, I can ask a question. Ali, you were frozen for a while and I took over, okay? So, and I'd like to take the opportunity to ask Rosenberg.
Go ahead.
It was very good. Oh, sorry. Can you hear me, Rosenberg?
Yeah, it was okay.
Okay. It was very good study. It was also complicated to comprehend. Too many things happening at the same time. No, too many things happening at the same time. I found it quite nice to see that this turbidity acting as a refuge for zooplankton, I guess. In the mesotrophic ones are the eutrophic, less eutrophic ones are in the dry region, isn't it? Less
Yes, both of them are they are in the semi-arid, but yes, the mesotrophic ones were in the less dry region, let's say.
Less dry. Okay. Now I'm with you. We also found similar results. I think it's very similar. If you have a wider functional feeding groups of fish, like piscivorous, omnivorous, and others, then it all distills down to the zooplankton as well, that you get wider zooplankton size diversity. And I think it's very nice. And in here, nutrient kind of plays the trick in terms of whether you have the piscivorous fish or not. That's really nice, your study. Yeah.
Thank you.
Yeah, I just wanted to make a comment. No question, actually.
No, that's okay. Thank you. Thank you very much. And I'm sorry, I'm asking sorry for everybody here if you could not understand my talk. And I did my best to make it as simple as possible. There were a lot of results. I also had the results of size evenness, but I decided not to add it because it would be too complicated. So I just decided to keep the size diversity ones. And just the last comment, Evaldo also asked about the size of the lakes. The lakes that we sampled, they were basically the same size. We tried to be careful with the sampling design to avoid this possible problems related to the size of the lakes.
Okay, Andros has a question, I think, for Rosenberg.
Okay.
Yes, sorry, I don't have my webcam on right now, but I hope you can hear me. It's actually a very simple question. First of all, nice work. I liked it very much. And I saw that you took this approach of comparing the two basins, right? The one that's more wet and the other more dry. And then I was wondering what would be the next step to really include a more landscape perspective to understand these relationships. Because as I see now, I think you put a lot of emphasis on this top-down and bottom-up perspective, which to me seems a little bit as kind of local processes. Yeah. Unless you can connect this variation really to the regional scale. So how do you envision this possibility, like to include a landscape perspective to, let's say, get a bit closer to what the session really proposed? I think this is not only for you, but for everybody, right?
Yeah, that's true. And actually, Ali, when we were discussing about this work, when I submitted the abstract, he told me that I should somehow make a relation because it was a very local process, like you said, Andros. We have some data for the land use, for example, when you're talking about the landscape perspective of this work. It's not a very simple question because we did a lot of work. We did a lot of effort for sampling these lakes, for example. And then, of course, that would be really nice if we could somehow increase the scale of this process, like you said. But if I understood what you're asking, we have some information about the land use as well that could somehow be related to the differences that we observed within the lakes and may not be related to only the precipitation differences between these basins. We know that in the semi-arid area, for example, in the more eutrophic lakes, the land use is completely different compared to the lakes that are inserted in the region that rains less. Even this is difficult for me sometimes to explain. But these differences in the land use may somehow be more important than the difference in the precipitation to explain the differences within the lakes. And we have these data sets, but we unfortunately didn't have the time to make this connection that you were asking for.
Nice, thanks, Rosenberg.
You're welcome.
Okay.
Sorry if I were too long in the answer.
No problem. But I think we should get moving on to the next talk, which is, we already discussed some of these questions previously in Wojciech's talk. So are there any additional questions for him? If there are none, I came up with one that I'd like to ask. So you guys did a latitudinal gradient, and you talked about cyanobacterial blooms, but I wasn't able to really understand the relationship between the latitude and the cyanobacteria. Did you just assume that there was more cyanobacteria in the southern lakes, or did you actually measure it and compare it to the zooplankton responses?
Actually, we counted our cyanobacteria under the microscope. However, we didn't present the data here because samples still need some processing. We have some delays due to COVID, and we are not able to present the straight data on the cyanobacteria biomass. We are operating with the fact that the cyanobacteria bloom was occurring, and we found that we were sampling the lakes during the cyanobacteria blooms period in all of the when the cyanobacteria bloom were present in each lake.
Go ahead, Meryem.
I was also wondering whether you had more fish in the south also. I mean, this latitudinal gradient you related with the cyanobacteria, but did you have a fish sort of, or did you have data on the fish to see some latitudinal gradient?
Actually, we didn't analyze the fish abundance, but we assumed that on basis of the our conversation with the authorities and of the lakes and the anglers which are present in the lakes, that there are fish in these lakes. Some of them are piscivorous, some of them are omnivores, and we assume that the fish predation pressure was on the same level in each of these lakes.
Okay, so I had another comment actually, because I thought it was very interesting the way that you guys classified the zooplankton according to latitude. Like, let me just open up my question here. The diet and the habitat. So, for example, we have lots of calanoid copepods in tropical lakes. Like, they're the main ones, basically, in terms of abundance and biomass. And I noticed that you guys considered them more restricted to higher latitudes. So it's interesting to see that there's all these assumptions about, you know, what the habitat, which people use as functional groups. So I think we still have a lot more work to come up with a unifying kind of a functional group matrix, because they might not necessarily agree. So that was just a comment on my part.
Yes, I think that we are able to, it is very hard to isolate each of the species we found in our samples and analyze the carbon to nitrogen ratio in the sample or the stable isotopes in the samples on the single species. And we have to make some kind of generalization functional character of each species, and we divided them into 4 groups, which are presented in the in our presentation: that calonoid copepods, cyclopoid copepods, Daphnia, and Diaphanosoma, which each of these 4 groups differs more or less in the functional traits and also the feeding habits.
Okay, thanks. Any other questions? No? Then I think we can move on to Nathan's talk. If anybody had any questions for Nathan. Is Nathan here? Hello?
Yes, I'm here.
Okay, hi.
Hi.
So if nobody had questions, I had one for you. I noticed that the pH also had a strong effect, and I wanted to know if the effect of pH was even more than that of total phosphorus for zooplankton diversity. And if so, do you think the results are driven due to agriculture, or they're simply driven by pH? Your microphone is off, Nathan. Hello?
Can you answer? Eu posso falar em português, você traduz para o pessoal?
I prefer not to.
Okay, but can you repeat the question?
Yes, so it seems like the pH was also an important factor, right? In addition to phosphorus. Is that correct?
Yes.
So if that's correct, then could the results be due simply to different pHs in the lakes? Because some of these lakes were very acidic. So if that's the case, then is it really agricultural nutrients that are driving the results, or is it the pH? I was just curious.
So I think I can help Nathan. I am Maria Estela, his professor. Nice to meet you.
Hello, that's fine.
Thank you for the question. Nathan is a little bit nervous because it's his first experience in a international congress.
Good for him.
Yes. Okay. So we believe that this is agricultural result, you know, because all the influence from the land use can interfere in the pH. So the pH is a kind of result of all the land use that we are also have some others influence from this kind of agriculture in our region. So I think it's agricultural.
I see. Okay, thanks.
What are the, I mean, I was wondering, what is the cause of such pH differences? Are some of those really acidic, like acid mines or something? Or it's only because of agriculture?
We believe that is because agriculture and some influence from nutrients that it's applied in this region. So influence the pH, and then because of this, we have results in pH and the nutrients also. And this is, we believe that this is very connected.
pH, then pH will be correlated with the nutrients, because I guess through the production, the pH increases.
Yes.
So there must be some correlation between them, autocorrelation between these two.
Yes, probably.
Okay, thank you. So I just realized that I'm
Thank you.
not following the order that I said we would be following. Too many things going on here. Sorry about that. I'm just following the random order that I wrote in my question. So we'll just keep going with that if you don't mind.
No,
So
no,
then,
it's your order, Ali.
yeah,
So
I'm
go
the
on then.
dictator here. So now we'll move to Sinem's talk, which is the title of her talk is Combined Bottom Up. No, hold on. That's Melissa's talk. Sorry about this. Okay, the
effects of dissolved organic carbon and zooplankton grazing on
thank you.
bacteria and ciliates.
Thank you. Now we have a democracy. Okay. So any questions for Sinem? Nestor had a comment for you, Sinem, for you and Melissa. He says that the experimental works on the interaction between DOC and herbivory are very interesting, and it's key to understand the associated effects of DOC in periods of heavy rainfall in Uruguay. But that's the only comment. So that's nice for you. I have a question. Even though I'm your co-author, I don't know if I'm allowed to ask.
Go on then, Ali. I am allowing you.
So you saw that there was very little herbivory, like top-down effect on bacteria. So the zooplankton weren't really connected to the bacteria, it seems. Do you think that the ciliates were responsible for this because there was some kind of a trophic cascade, like the zooplankton were eating the ciliates, so there weren't enough ciliates for the bacteria? Do you think something like this is happening?
Actually, I am really confused about that point, but I thought in two ways. First, one is both bottom-up effect and top-down effect neutralize each other. I mean that the DOC enhance bacterial growth, but on the other hand, grazers continuously eat them, and they make them constant biomass. This is the first thing. The first idea I thought about. And the second one is I showed the final results at the end of the biomass results at the end of the experiment. And 4 days is too long period for to see DOC effect on bacteria, because DOC is metabolized by bacteria in 24 hours. In the literature, I saw that, and this may be the reason.
I see.
So Ciliates somehow hijacks the food from the from zooplankton.
Actually, bacteria has lots of enemies: HNF, Ciliates, Daphnia, rotifers, etc. And all of these consumers eat them, so they are very unlucky.
Yeah. And I just wanted to make another comment just for every one of us here. Sinem's work, I mean, it's a little bit self-promotion here, but it shows that, you know, Daphnia, we often consider them as herbivores, but they are very happy eating ciliates. They eat quite a bit of ciliates. So for all of us who think about, you know, how to categorize, how to make that functional diversity matrix, right? We want to keep adding some, let's just add the diet here too, because we have more our model work better then. So we have to be really careful with assigning these things just based off of assumptions, because they might not be true. So that's just a reminder for all of us here.
Yeah.
I have an additional comment.
Please go ahead.
Yeah, I like very much both dissertation about the interaction between dog and grazing pressure. And I think it's quite important to start to incorporate a very simple proxy like the watercolor when we analyze or compare different shallow lakes. It's a very, very simple proxy like Sekidep, but we never pay attention and the gradient of the organic dissolved matter, and of course the organic carbon play a key role in several shallow systems in South America. Not only the shallow system located in the floodplain, also in the.
Can I say something? I think I somehow understood because it was also Nestor, it was also my concern. Like if you asked me to do such an experiment 5 years ago, I would have laughed at you, because we wouldn't have such DOC problem in Turkish lakes. It's all warm and we never get that really, we get a regular pattern of rains and stuff. But now,
Meryem, many people couldn't understand because Nestor had a weak internet connection. So I'm going to ask two things. One, please, if you're not talking, mute yourself. I'm hearing some laughter, which of course it's fine to laugh, but it interrupts. And many people, including myself, couldn't understand what Nestor said. So maybe he can.
Okay, maybe repeat it.
He can repeat it, because I think he's back now. Or if he's not back, Meryem, maybe you can do a recap.
Okay.
What I understood that how critical DOC is, but Nestor is, if he's back, he can sort of. Nestor, can you repeat what you just said earlier, that we couldn't hear you well because of a bad connection?
Okay. Sorry for the bad connection. I'll try that again without the video, with the camera. I think it's very important, the experiment that analyzed the interaction between dissolved organic carbon and the grazing pressure. And I'm thinking that we must promote when measure critical or relevant attributes in shallow lakes, include also a very simple proxy like the watercolor that we never pay attention when we compare a group of shallow lakes in the landscape scale. And it's a very simple, and in several parts of America, it is an incredible gradient in terms of the dissolved organic matter. And we must pay attention because it's quite simple, like the Sekidep or other limnological classical proxies.
I think I understood well. This is becoming a must now to focus also on the DOC because of these extreme events. You know, the extreme rainfalls that even in Turkey, we have this problem lately. I mean, like Ankara is a cold, dry area that we never used to have such extreme rainfalls. But now it's happening with a high intensity and those things, those heavy rainfalls in the catchment brings a lot of DOC to the lakes that I think we cannot avoid it through the climate change. That was the idea how it came about on this experiment. Otherwise, I wouldn't have thought about it like 5-10 years ago, doing such an experiment or planning to do such an experiment in the scheme of aquacosm. Yeah, I just wanted to add that I agree with Nestor. It is really important to know the DOC levels now.
Okay, so let's move on with my random order that I created for myself, and I'm imposing on everybody here. So the next talk will be Lorena. Could you please repeat the title of your talk, because the system I created made it very difficult for me to navigate myself. Lorena, are you there?
Are you hearing me?
Yes. So could you just
Okay.
repeat the title of your talk?
Okay, it's trait-based zooplankton community assembly and the functioning of aquatic ecosystems along a productivity gradient. Did you hear me?
Yes.
Sorry,
Just,
okay.
just waiting to see if anybody had any questions. So I guess now people are shy.
My co-author have a question.
I think Rosenberg has a question.
Yeah, so we can start with Rosenberg.
Okay.
Okay, thank you very much for your talk, Lorena. It was very interesting. I really like it because it's quite related to what I did as well. So actually, I have a very simple question. Your experiment did not take into consideration the fish. So I wonder if you can actually see this type of pattern in the real world. You somehow saw in my in my work. So I just would like to hear from you if you can actually generalize the pattern that you found in your experiment.
Yeah, this is an interesting point. Here in our country, in Brazil, in the subtropical and tropical regions, we could not see this type of scenario, I think, without fish pressure. It's difficult. But as my co-author, Andres Gianluca, said in my thesis defense, this is commonly found without fish pressure, fish predation. It's commonly found in Europe, this type of ponds. And besides of that, okay, if we cannot generalize like this experiment for the world because it's difficult, it's hard to imagine a lake without fish pressure. I think it is important experiment that show this type of pattern without cyanobacteria blooms and without fish pressure to disentangling the effect of direct effect of eutrophication from the indirect effects of eutrophication, which means high fish pressure and also cyanobacteria blooms. So it is important to understand the real effect of nutrient addition. That's for me, it seems to be different of the effect of cyanobacteria blooms and fish pressure, for example. So, okay, if we cannot extrapolate these results for the most of lakes, we can at least understand the effect of each factor, okay, in biodiversity and also ecosystem functioning.
Okay, thank you.
Good answer, Lorena, because whatever you do, somebody's going to ask that question. If you have the fish in, they're going to say, well, then how do you know what the mechanism is? And if you don't have the fish, they're going to say, well, how is this relevant?
Yeah, exactly.
So it's not which one, we need both, I think. So that was a good answer. I think
just a compliment that I and my co-authors, we recognize that fish predation and cyanobacteria are of key importance for the size change of zooplankton, okay, especially along a productivity gradient. But as I said, it's really complex, disentangling the effect of all factors that eutrophication can cause. So what we did here is important, for example, to evaluate lake restoration effects, such as nutrient loading reductions, what is happening, especially in temperate systems, rare eutrophication, and also to support management decision-making. So,
yeah. I have a quick question. How did you manage to add nutrients and not get cyanobacteria in the mesocosms?
yeah, this is a trick question for me because I didn't perform this experiment. This experiment was performed by Steven in 2005, if I remember correctly. So we, in fact, we don't have the phytoplankton data to see, but as Steven told me, there's no like cyanobacteria blooms in the tanks. They didn't find cyanobacteria blooms in the tanks. So
you got lucky.
Yes.
Okay, thanks. Andros, I'm gonna have to skip you because we're running out of time and we still have quite a bit of talks, but if you want, I'm sure you can ask Lorena. Sorry. So the next talk on my personal list is Vildan. Is she here?
Yes, I'm here.
Hi.
Hi.
Could you please repeat the title of your talk, please, for everybody?
Okay, my title is the Phytoplankton Community Assemblage Processes of Turkish Shallow Lakes Using Different Functional Diversity Components.
Okay.
It's a long title.
Questions for Vildan? Anybody? Go ahead, Rose Rosenberg.
Okay, I was just wondering, it's a very quick question. Can you actually say that there is a relationship between functional diversity and total phosphorus and total nitrogen in your work? Because when I see the graphs, they don't seem to be related. I mean, you have a lot of points, and probably there are some outliers there. So if you remove the outliers, could you actually say that there is a relationship between phosphorus and nitrogen and functional diversity?
Okay, actually, it is the first I removed the outliers, and this is what I have left, and still not very strong interaction, I know, but we think that it is because the data in the data set we almost have completely lakes within the eutrophic state. So there was not a large interval for the phosphorus and nitrogen, and I think that's why that we don't get a better result. And now we are trying to try with different lakes with different conditions, and we want to try the same process with the lakes that we have the time series data. So eventually we are hoping to find a better result. So I think that, okay.
I had a quick question for you. It's more of a general question. Is dispersal only limited to geographical distance, or could topographical barriers like mountains be also an important limit for dispersal? Because I noticed that you only consider geographical distance, but it could be something else too. What do you think about that?
I also tried with the topographical differences. Because in the data set we have that we have lakes that are in the highland regions and the lowland regions, and I also tried them to see if there is a difference between the highland and lowland lakes, and I couldn't also find any relationship between them with the composition and their locations. So we went on with the geographical distance. But we are also trying to do a more complex distance decay analysis. But this is an ongoing research for me, so hopefully, yeah.
Okay, sounds good. Lorena has a question, I think.
Okay. Yes. I have the same point of view of Rosenberg, and I was thinking about the absence of relationship between your community and abiotic environmental variables. And maybe it's not a question, but it's just a suggestion. Maybe you can check the relationship with nutrient ratio. Because all the lakes, as you said, they are eutrophic, but maybe the ratio between nitrogen and phosphorus are not common between them, and also other abiotic environmental variables like euphotic zone deep, I don't know, light can be a factor. And also, just one question: did you try mixed effect models with your lakes as a random effect? Because it can be another thing that you can try to see if there is any relationship between.
Yeah, we only use the generalized linear models, but maybe this is why we don't get any bad results. So maybe we can try another modeling. And thank you for the suggestion.
Not just because I also it was also hard for me like find the relationships between the community and abiotic variables with a big data, and maybe mixed effect models work for me. So maybe it's something that you can try. And I also saw that you put some future directions, and I think that you should check the biodiversity relationships, that because maybe between your variables, biotic variables, you can find a really nice relationship.
Yeah, thank you. Thank you. We will try that. So, thank you.
Just a small addition. Those lakes are shallow lakes, so, but I think you try Ciliates, didn't you, Vildan? We tried all the variables that we measured, including Ciliates and zooplankton biomass, fish, different fish feeding groups, you know, piscivorous fish, young of the year fish, omnivorous fish, everything. It's because that was the data was produced through the snapshot sampling of lakes for everything, macrophytes, even the sediment cores and everything. But they did not come out, but we didn't try the ratio. Maybe that would be good. Some of the lakes are nitrogen limited. Maybe that might come up something there as well.
Yeah.
Thank you, Lorena.
Yeah, thanks. So, yes, we may need to determine the time for in the next sessions. Okay, that's anyway. So I think we should get going to the next speaker. Let's go with Melissa. Her talk is on the combined bottom-up effects of DOC and top-down effects of zooplankton with contrasting traits on phytoplankton biomass and composition.
Yeah, thank you. I'm glad to be here.
Hi.
Hi. Can you hear my voice clearly?
Yes.
Okay.
Any questions for Melissa?
Go ahead, Rosenberg.
Yeah.
Okay, I'm sorry, I think.
That's good.
Oh, well, if nobody's asking.
No, you go ahead with it. Okay, great.
Okay, Melissa, thank you very much for your talk. It was very nice.
Thank you.
Well, I'm just in doubt about the reduction of Cyclotella by Copepod, for example. I think I may not have understood it, but I want you to confirm if the predation was the same in respect of the treatments that you are using with DOC or without DOC. If the effect of the copepods on Cyclotella was the same. I could not actually see that. I'm not sure. Probably the statistical analysis are saying that actually there are some differences. That's actually my doubt.
Yeah, thank you for question. I guess the copepods are selectively feed on Cyclotella because of their nutrition quality and their size, because they are larger compared to other species. But as you said, there is a kind of not significant, let's say, but there is a difference between with DOC and without DOC. And what we see in the result that when there is a presence of DOC, copepods more feed on Cyclotella, let's say, compared to without DOC treatment.
Thanks.
I have a follow-up comment on that. So, like following up the same idea, wouldn't you expect, because Cyclotella is bigger in general, that other zooplankton would also just eat it more, not just copepods? Because copepods are selective in general, right? So if it's bigger for them, they're going to find it easier, they're going to eat what's there. But also, like, Daphne is not selective, but if the prey is bigger, they're also going to eat more. So why do you think you didn't see that as well?
Yeah, actually, we see that both Daphne and copepods significantly eat Cyclotella and decrease their biomass. But there is some kind of more copepods feeding on them, let's say, because we related with this is the selection issue. But you're right, both of them feed on it.
Okay, thanks.
And in general, if I remember correctly, copepods grazed more than Daphnia, regardless of the prey type.
Yeah, exactly.
So that might also be.
Which is very cool, actually, because it also like confirms some of the patterns that Ali showed in his presentation as well.
Welcome to the Copepod Club.
I'm becoming a copepod person as well, slowly. Still a Daphnia person.
maybe next Shallow Lakes, I'll make a copepod session.
Yes, definitely. Your community is building up.
Thanks, everybody, for the questions. I'm just going to keep pushing us forward because we technically have 2 minutes, but we're not going to be able to finish. I hope you can hang on until the end of our session. We only have 3 more talks left. So let's go with Mary's talk, which is about the flower litter. Is Mary here? Her talk's titled Flower and Leaf Litter Interaction Promotes the Non-Additive Effects in Aquatic Environment.
I think she's not here. She was at the beginning, but not anymore.
Oh, that's too bad. Do people have, are our emails available to each other? If not, I'll make sure that they are. Melissa said they are. Okay.
They are written in the book of abstract, I guess.
Yeah. So if anybody has further questions to Mary, it can be, yes, Evaldo, you can go next. And then there's just one after you. So the floor is yours.
I mean, should I say the
title?
Oh, okay.
Yes.
Right. So the title of my presentation was Importance of Copepod Grazing and Cyanobacteria Defense Traits on Top-down Control of Cyanobacteria.
Questions for Evaldo? I know we're at the end of our session, but we're almost there. Evaldo's actually the only one where I didn't come up with any questions because he already published everything.
It was a great talk. I mean, I must say that it was a great talk. Really nicely presented and the excellent results.
Thanks, Mary. Yeah, I got lucky on the experiments.
Really lucky, excellent results. And a really nicely questioned and hypothesized. Very good. Yeah.
Yeah.
Thanks.
Well, I guess
move
for
on.
what?
If there are no questions, we can move on to.
I have one question. Just one doubt. Your presentation was really nice, Evaldo, and I love your works. Also, just one doubt about, in the end of your presentation, you said that feeding traits and prey defense traits are important to understand the impact of plankton biodiversity on ecosystem functioning. And what are the prey defense traits? Because I didn't follow, I didn't get during the presentation, what type of traits did you mean prey defense traits?
Sure. So when I talk about like both grazer traits and prey traits, the grazer traits are pretty clear, which are the grazing modes. And the prey traits, in this work particularly, I address by using two cyanobacteria species. So Microcystis is, when cultured, is usually single cell but has microcystin as a type of toxic defense. Whereas the other cyanobacteria, Raphidiopsis or Cylindrospermopsis, is a filamental, so it has the shape, the different shape, which can be a deterrent by itself, but also has a different toxin profile, which is Cylindrospermopsis. So those two are different from the defenses that the microcystis show, and these may represent this like difference defense traits.
Okay. And do you think that those traits are related to body size, like the size? Sorry, not body size, but the size of the prey?
Actually, that is a very good question, but kind of tricky, because the microcystis itself in nature, it's usually in like colony. So it's like hundreds of cells in a colony with a mucilage around it. So there's all of these extra morphological features that grant them extra protection. So, you know, if we see this kind of like grazer deterrence just by single cells, I would assume that in nature, where microcystis is presented as colonies, the level of grazer deterrence might be even stronger. So the combination of the fact that in our experiments, it's just the toxin difference, but also in nature, you have the morphological aspect, may lead me to think that Microcystis is even more avoided by copepods than other cyanobacteria groups, such as Ciliates.
Okay,
Size does play a role, but in our experiments, we couldn't assess the full potential of size as a prey defense because of this particular aspect of Microcystis.
Yeah, because I'm just thinking about what Rosenberg said about his results, that he didn't present this result, but he said that size diversity of phytoplankton, of the prey, is correlated with size diversity of zooplankton, for example. So,
I see. Yeah, it would be cool to see like, I'm more in the experimental side, so I don't recall reading anything about enrichment and, for example, colony size, which can be an interesting question, interesting data that could address your question. So if we have more like nutrients and the colonies of like cyanobacteria in general, or the filament size increases, then you could have more of this like nutrient size diversity within cyanobacteria.
Yes. Okay.
And that would definitely be something interesting to see.
Yes, it is. I am a size girl, but Ali knows that. But it's okay. Thank you for your answer.
Thanks for your question.
Okay, thanks, Evaldo. Were there more questions for Evaldo? Okay, so we've come to the last presentation, which was from our group, and the title was Zooplankton Grazing Mode and Body Size Predict Ecosystem Function Across 16 Tropical Lakes. Let me scroll up here. I remember that is Nestor still here? Yes, he is. So he said that it's remarkably interesting. Thank you. And a great challenge is to couple a good functional strategy for zooplankton with an adequate functional strategy for phytoplankton when we can explore the grazing pressure. What alternatives do I see? So I'm not sure what you mean by functional strategy. Could you please explain that just so I can understand more?
Yes, of course, Ali. Exists several functional approach for analyze the phytoplankton community, mainly for the prediction of the possible assemblage of different group or different species according to the environmental drivers. But I think it's very interesting, Ali, try to explore possible functional approach of the phytoplankton community from the perspective of the grazing pressure. Of course, the size, the presence of toxins are critical or relevant, but I think we must think both functional approach between the prey and the predators at the same time. And in this sense, I like very much your dissertation, Ali, because I think improve the classical traits that we pay attention to the zooplankton community.
Yeah, thanks, Nestor. So you're right. I think the metric that we measured, the function, the resource use efficiency, it's just the biomass ratio of the zooplankton to phytoplankton. So that already implicates that we need to look at both of the traits. So it's a nice function to study because it links the producer and the consumer. And we did have some very interesting regressions between, for example, different morphology-based functional groups of phytoplankton and distinct zooplankton functional groups based on grazing mode, for example. So we saw that, if I remember correctly, I think it was the filamentous cyanobacteria had a really nice positive relationship with the cyclopoid copepods, for example. So looking at these kind of traits like producer-consumer trait relationships, I think would be a good place to start to try to understand if this is by some kind of trophic interaction or an indirect positive interaction or just coincidence. But I think that would be a good place to start. I would start with, I mean, we are planning on doing this. I just didn't have enough time to show it in the 15-minute presentation. But we're planning on doing this with the morphology-based functional groups because with the Reynolds classification, there's just too many and it doesn't really become effective when we're trying to do what we're doing. So that's what I would suggest. That doesn't include toxin production. So it's not going to be perfect, but that's what I would answer. That yes, we should definitely look at the two together and we could start pairing traits.
Maybe, Ali, I think that the scheme of Carla Krug, the morpho-functional group of phytoplankton, maybe is a good alternative for exploring.
Yes, yes, that's the one that we're using. That's
Ali,
the one that we're using.
Just as a, continue what you're saying, it's Evaldo here. So you say that the Krug morphological functional groups don't necessarily address toxin production or toxin content. But can't you just tweak at your will to since we have the knowledge that, within the phytoplankton community, there are some filamentous phytoplankton that produce toxin because they're cyanobacteria and some colonies that produce toxin. So can't you just like make subcategories in those, since you have this information from your phytoplankton community data, and then try to see relationships of like filamentons, non-toxic and non-toxic? Like, is it possible? I don't know if it's possible.
I mean, of course it's possible. It just doesn't mean that necessarily just because you say that, okay, I'm going to define microcystis as a toxin producer, that it necessarily is producing a toxin. So we can create a group saying potential toxin producer, for example. Yeah, definitely. That might actually, I think the Carla Crook's functional group has been updated to include an 8th group. I can't remember if that was related to toxins or not, but yeah, definitely that's an idea. It's just that a lot of the times the same species that can produce a toxin someplace might not be producing it in another place, or it could be producing more toxin or altogether different variants. So then it would be, I mean, we can do it. It's just how much does that really represent reality?
Yeah, no, I totally understand. Thanks.
Thank you. Andros has a question. No, I won't skip you, Andros. You're not my advisor.
Okay, so thanks. Yeah, so although I'm a co-author in this study, I think it's still preliminary, so it's more like a question or comment in order to further reflections or discussions. So I think, so one of the main results seems to be that body size mean is important, right, for grazing pressure. But at the same time, you have the dominance of cladocerans being also important, right?
Yes.
So how do you reconcile this? Because in tropical systems, cladocerans are actually much smaller than the copepods. So my question or comment is more like, if you subdivide the data and look exclusively on cladocerans, should not this relationship between size and grazing pressure get even stronger, just as an exercise? I'm not saying to throw away the copepods entirely, but to dissect, I don't know, yeah, to dissect more this relationship between size within cladocerans to see if it gets even clearer. Because you have a lot of large copepods that are not so efficient, I think.
So that's a good question. And I showed in my presentation the best models. We tested all kinds of regressions. So the interesting thing about the size and the top-down pressure was we found it also for calanoid copepods. We did not find it for cyclopoids, for example. So even though cladocerans were like 5% of the total zooplankton biomass, it's extra interesting that those were the best top-down grazers in the system. So that to me indicates that there's really something else other than size that's important, which is, I think, the way they eat, which relates to your second question. What's going on, like how can copepods be, like maybe copepods are less efficient, I think you said, right? So it does seem like even though they're smaller sized, and they're not actually that smaller, when we compare the mean size of the cladocerans and the copepods, they're not that different. And that's because most of the biomass are the juveniles. They're not the adults in these systems. So there's lots of copepodites. So their size might actually be even smaller. But no, it's a good suggestion. And I will separate the data just for cladocerans to see if the size effect changes among the different groups of zooplankton. That's a great idea. Thanks. Rosenberg?
Yes, it's also a quick question, Ali, and probably the last. Well, you said at the beginning of your talk that quality is very important for copepods. And why do we see in our results, for example, a strong relationship between copepods and cyanobacteria, for example? I was just wondering, like you said, I think a few minutes ago, that maybe it's just a spurious correlation, for example. Maybe copepods are increasing because there is another factor that may actually be benefiting copepods. Yeah. So I just would like to hear a little bit from you about that.
Well, I think Sinam's talk has some clues for this, because she showed that the copepod ciliate link is really strong. And one thing that we know happens during cyanobacterial blooms is that the microbial loop gets more important. So there's a lot more detritus in the system that's feeding, ciliates, bacteria, whatever. So the copepods are probably relying on that instead of the cyanobacteria. But since there's more cyanobacteria, they're benefiting indirectly. And that's why we see the positive interaction between cyanobacteria and especially cyclopoids. With calanoids, we didn't see any relationship between cyanobacteria biomass. So I think that's the main point, is that they're just eating non-phytoplankton during blooms.
Thank you very much. And I forgot to say that your talk was very nice.
Thank you. Thank you for making it possible, the study. And so Evaldo has, okay, I'm going to stop. This is going to be the last question because I have to teach a class in a few minutes. And before everybody starts leaving, I just want to remind all of the early career people here that we have the early career workshop. So please remember to join in a few moments. So Evaldo's question is the last question. Thank you, Nestor. Evaldo's asking, how much do I think the eco-evolutionary process contributes to the copepod-cyano relationship? I think quite a bit, Evaldo. I mean, we know that from your work too, because since we didn't find any relationship between cyanobacteria and copepods, that means that they're not really benefiting from them, but they're not also grazing them that strong, but they are grazing them. So if they're able to be there and dominate during blooms, by definition, there has to be some kind of local adaptation there. Whether or not that's just the grazing avoidance or it's some physiological tolerance, we can't say. Okay, everybody.
Ali,
yes.
Ali, can I? Yeah. I think
yes.
It was a very, very good talk. Your talk is, I think it will also kind of, in a way, to make some shift in our understanding as well. So it might be a very cornerstone. I hope that you will publish it soon. If I'm not mistaken, I watched it last night. Actually, this morning, early 2 in the morning or something.
Oh gosh, nobody deserves that.
Sorry about that. It's not that I don't value any of your talks. I mean, in general, our group, but I was sort of not able to make time before. I was thinking that within the trait identity, how about the intraspecific variations? I don't know, maybe it was in the midst of the early morning that I'm confusing things, but like having the wider niche, would it make differences?
Yeah, that's an interesting question. I mean, size kind of tackles that when we're talking about intraspecific for size, but in the other ones, like Evaldo's question, it relates to the toxin tolerance, for example. There might be differences in toxin tolerance among species, but then that's a weak spot. That's a huge gap, I think, in the literature. We just simply don't know. Yeah, and that's the big danger of assigning these traits.
Yes, yeah, too much of a generalization, and definitely you're right that with size we know the niche width, based on the size, but within the identity also there might be from the eco-evolutionary perspective as well. that I thought maybe in future the direction would be also what you brought up in your talk, as well as within trait identity variations. I don't know.
Yeah, I think we need some more experiments, some microcosm experiments.
Yes.
For that.
Small bottle experiments that I value now.
Experiment.
Okay. I think it
But
was a great session.
Meryem. Yes, people, thank you very much for your presence and your participation. We approximated as much as we could the real experience of being together with colleagues and friends, and we miss you all, and thank you.
Yes, thank you very much. I think thank you for joining us.
Definitely.
And it was a
Meryem.
great
And hope you enjoy
collective effort. Yeah.
Yes, and hope you enjoy the rest of the conference.
Yes.
See you around.
Yeah. See you around. Bye-bye.
Bye-bye.
Not on the beach though,
Bye.
I'm sorry.
Next
Could have
time.
been nice to be on the beach to bump into any of you, but yeah. Okay, bye-bye.
Bye-bye.
Bye.
Bye bye.